Hello everyone. In this class, we'll try to understand what are the different properties of cardiac muscles. So the properties of cardiac muscle, that is electrical and mechanical properties, are where electrical properties are excitability, which is called as bath- bathmotropism, autorhythmicity, chronotropism, conductivities, dromotropism. Whereas mechanical properties are its contractility, that is inotropism, and other properties like refractory period and Frank Starling law. Excitability. The cardiac muscles are excitable and respond to proper stimuli with the production of action potential. In heart, two types of tissues are present: the conducting tissue and contractile tissue. The conducting tissue, they initiate and conduct impulses, that is SA node, AV node, internodal duct, bundle of His and Purkinje fibers. Whereas contractile tissue, they contract in response to impulse conducted uh, in myocardial cells forming walls of the cardiac chamber. Action potential can be defined as a sequence of changes in the membrane potential in an excitable cell due to opening and closure of different ions after the application of threshold stimulus. And action potential in cardiac muscle is of two types: fast response action potential and slow response action potential. In fast response action potential uh, is seen in working myocardium, that is atria and ventricles and Purkinje fibers, and it is characterized by rapid depolarization. When the firing level is reached, there is a very rapid change in the membrane potential, producing a sharp upstroke which is called as zero phase. The resting membrane potential of ventricular musculature is about minus 90 millivolts. In this phase, the membrane potential crosses the zero line, that is positive overshoot. The inside of the cell becomes positive with respect to outside, and the cell is fully depolarized. The initial depolarization is due to sodium influx through rapidly opening sodium channels. In initial rapid depolarization after the zero phase, uh, there is there is a slight fall of membrane potential towards zero potential line, and this is named as phase 1. This phase is due to closure of fast sodium channels and opening of potassium channels. That means efflux begins here. Then we have a plateau phase. After phase 1, the membrane potential is maintained for some time at that level, that is, the repolarization is prolonged. This phase is named as phase 2. It is due to the opening of slow calcium channels along with potassium efflux. Thus, the potential change due to the potassium efflux is neutralized. Late rapid repolarization. When there is a rapid repolarization at phase 3, this is due to the closure of slow calcium channels and potassium efflux. Baseline, that is the polarized state, is the last phase where the RMP is restored by the activation of sodium potassium pump, which is caused by potassium efflux. So these are the different phases of action potential in the cardiac muscle. There is a zero phase which occurs because of the rapid sodium influx. The phase 1 occurs because of the potassium efflux, and the plateau phase occurs because of the continued potassium efflux and long acting calcium channels, entry of long acting calcium channels. And again, phase 3 occurs because of the continued potassium efflux. Another action potential is slow response action potential. This type of action potential is seen in the pacemaker tissues. Before the sharp upstroke, which is seen in myocardial cells, there is a gradual slope here. In sinoatrial node and AV node, the depolarization in the beginning is gradual and RMP is automatically approaches to minus 60 millivolts. This phase of gradual slope is called as pacemaker potential. When this pacemaker potential reaches the value of minus 60 millivolts, firing occurs. Now there there develops a sharp upstroke and there is no plateau in action potential of the SA node and AV node. The ionic basis of slow response action potential is at the peak of each impulse, that is end of the depolarization, potassium efflux begins efflux brings about repolarization. Then there is a gradual decrease in potassium efflux followed by the opening of transient calcium channels and there is calcium influx, and this accounts for prepotential. When membrane potential reaches the firing level, there is opening of long lasting calcium channels and there is a calcium influx. When the membrane potential reaches the plus 35 millivolts, there is a closing of calcium channels and opening of potassium channels. Note that electrical and mechanical response overlap in cardiac muscle in contrast to skeletal muscle. This diagram showing the action potential of slow response type of action potential in AV node or SA node. So the initial phase is not rapid but it is very slow, which is called as a pace pre-potential till it reaches the threshold level. The ionic basis is for the pre-potential. In the initial part it is because of the delay in the closure of the potassium channels, that is closure of potassium channels. Then the transient calcium channel which causes the entry of calcium, and then there will be development of the threshold, so which leads to long acting opening of the long acting calcium channels that leads to the depolarization, and the repolarization is caused by potassium efflux. Autorhythmicity, the property of spontaneous prepotential followed by action potential, is called as autorhythmicity. The resting membrane potential of working myocardial cells is about minus 90 millivolts and is stable. In conducting tissue, resting membrane potential is about minus 60 millivolts and it is unstable. Because of continuous change in the membrane permeability, their membrane potential declines towards the firing level spontaneously after each action potential. When the firing level is reached, an action potential is triggered. This slow depolarization between action potential is called as prepotential or pacemaker potential, or also it is called as diastolic depolarization. The ionic basis of prepotential, as we discussed, at the peak of each impulse at the end of depolarization, potassium efflux brings about repolarization. Then there is a gradual decrease in potassium efflux followed by opening of the transient calcium channels. Effect of vagal stimulation on membrane potential. When cholinergic fibers to nodal tissue are stimulated, heart rate decreases. Acetylcholine at the nerve endings increases potassium conductance at the nodal tissue, which are mediated by the M2 type of muscarinic receptors. There is an increase in potassium efflux at nodal tissue, resulting hyperpolarization. In addition, the activation of M2 receptor slows the opening of calcium channels as well. This results in decrease in the firing level. Here you can see that the space between two pacemaker potential is going to increase with the stimulation of the vagus and the heart rate decreases. Effect of sympathetic stimulation on membrane potential. When sympathetic cardiac nerves are stimulated, heart rate increases. Noradrenaline released makes membrane potential fall more rapidly, which is mediated by beta 1 type of receptors. Noradrenaline facilitates the opening of calcium channel and thus rapidly depolarizing. It rapidly increases the depolarization phase. Note that discharge frequency is increased when the temperature rises, and This is going to explain the tachycardia associated with fever. Okay, so here you can see that with the sympathetic stimulation, the distance between two pacemaker potential is going to decrease because of the rapid depolarization. This diagram shows the difference between the action potential and mechanical response in a cardiac muscle where they almost overlap each other, whereas in skeletal muscle the electrical the action potential occurs quite early compared to the mechanical activity which happens later. Digitalis depresses the nodal tissue and exerts an effect like of vagal stimulation, particularly the AV node. The rate of impulse production in SA node is around 70 to 80 per minute, AV node it is 40 to 60 per minute, in bundle of S it is 30 per minute, in Purkinje's fiber 15 to 40 per minute, atrial muscle 40 to 60 per minute, whereas in ventricular muscle it is 20 to 30 per minute. Conductivity. Conductivity is a property which enables the myocardium to conduct electrical impulses is called as conductivity. Human heart has a specialized conductive system through which the impulse from SA node are transmitted to all parts of the body of the heart actually. The conductive system of the human heart comprises of SA node, internodal pathways, AV node, bundle of S and Purkinje system. Contractility is the ability of a tissue to shorten in length, that is contraction, after receiving a stimulus. Isometric as well as isotonic contractions are seen in heart, and factors that affects the contractility are the Vmax. Vmax is the velocity of contraction is maximum when afterload is 0. The maximum velocity which will develop when the afterload is 0 is called as Vmax, and it is increased when contractility is increased, and it is reduced when contractility is reduced. It is not changed when the initial length of the muscle fiber is changed. Note that the preload, the degree to which myocardium is stretched before it contracts, and is equal to end diastolic volume. Here the load acts before the muscle starts contracting, and afterload is nothing but resistance against which the blood is expelled from the heart. Here the load acts on the muscle after the muscle has started contracting. So this diagram showing the relation between the Vmax and the afterload. So when afterload is 0, the Vmax is maximum, that is velocity of contraction, and as afterload increases, the velocity is going to reduce. Extrasystole and post extrasystolic potentiation. In regularly beating heart, if an extra stimulus is delivered so that it falls earlier than the usual timing, but after the refractory period, then the heart responds to this stimulus by weak extra contraction called extrasystole. Now, the next regular impulse comes, but it finds the heart refractory due to extrasystole. So there is no contraction, and the gap produced is called as compensatory pause. Subsequent regular impulse comes and the heart responds to it stronger than the normal contraction due to the post extrasystolic potentiation. The cause for this post extrasystolic potentiation is increased availability of the intracellular calcium. Refractory period. It is the period in which the muscle does not show any response to a stimulus. Normal refractory period of ventricle is around 250 to 300 milliseconds, and that of atria is 150 milliseconds. A refractory period is of two types, absolute and relative. Absolute refractory period, where it is around 180 to 200 milliseconds. Absolute refractory period is the period during which the muscle does not show any response at all, whatever may be the strength of stimuli. During phases 0 to 2 and about half of phase 3, the cardiac muscle is in its absolute refractory period. Relative refractory period is for about 50 milliseconds. The relative refractory period is the period during which the muscle shows response if the strength of the stimulus is increased to maximum. It remains relatively refractory period until the phase 4. Significance of which is, since the absolute refractory period extends throughout the contraction period, the following cannot be produced in the cardiac muscle, that is the summation of contraction, tetanus and fatigue. So this diagram shows the action potential and the myocardial contraction, where you're showing absolute refractory period and relative refractory period. In absolute refractory period, the second stimulus will not be able to produce any response, whereas during relative refractory period, the increased strength of stimuli sometimes can evoke action potential. Frank-Starling's law. This law states that force of contraction of the heart is directly proportional to the initial length of cardiac muscle fiber within physiological limits. Initial length of the muscle fiber is equal to length of the fiber at the end of diastole, that is end diastolic fiber length. It may also be expressed as end diastolic blood volume. When the ventricle is in early diastole, it is more compliant, that it is more distensible. The blood at this stage enters the ventricle, but the tension, this is called as passive tension of the ventricle, does not rise appreciably. Only when the ventricle is reasonably filled, from then the passive tension is going to begins to rise steeply. The developed tension increases as the diastolic volume increases until it reaches maximum, and then tends to decrease. That is the descending limb of Starling curve. The descending limb limb in human heart is seen only when the cardiac muscles are ruptured. This law is an important role in the regulation of stroke volume and hence the cardiac output. So this is diagram showing the passive tension which is raised because of the diastolic filling, and so this the tension is developed, the total tension because of the contraction of the muscle fibers, and the difference between these two is called as the active tension, which is occurs because of the only the contraction of the ventricular muscle mass. Physiological significance of Starling's law. This law explains why blood is ejected by each of the ventricle per beat is same. The right ventricular output is more than left. Blood accumulates in the left side, leading to greater force of contraction of left ventricle, thus increasing the left ventricular output. It serves as a life-saving device in cardiac failure. Left ventricle failure causes accumulation of the blood in the left ventricle, thereby decreases the blood supply to the vital organs. Soon, accumulation of the blood in the left ventricle increases the initial length of the muscle fibers, leading to greater cardiac output according to Frank-Starling mechanism. If accumulation of the blood exceeds the physiological limit, the law fails to operate, leading to death. When peripheral resistance is increased, initially the heart is unable to pump all the blood it normally does. The accumulated blood in the ventricle stretches the muscle fibers, leading to greater contraction, and thus stroke volume is restored to normal in spite of greater resistance to the flow. Some of the factors which increases the end diastolic volume are increased pumping action of the skeletal muscle, increase in the blood volume, sympathetic discharge, increased intrathoracic pressure, that is thoracic pump, whereas factors decreasing the end diastolic volume are sitting and standing posture, reduced total blood volume, decreased ventricular compliance, and increased venous resistance, which occurs in pericardial effusion, and increased intrathoracic pressure during respiration. So this is in brief about the properties of cardiac muscle fibers. Thank you.